started. Thank you all for coming, for getting up so early to hear more about the prevention of infection trial results. We really wanted to be able to give you all an opportunity to be sure that you were comfortable with the results, that you understood them, that you had a chance to ask questions about the trial, about the data, about implications and next steps. So we're going to start this morning first with Mark Catherall, whom I'm sure you all know, but director at SATFI and at the University of Cape Town, and importantly, principal investigator for this trial. He's going to do a brief recap of the results just to make sure everybody's on the same page before we start with questions. Then we'll do a bit of a moderated panel up here to try to get out some of the main themes and what we think may be burning questions on people's minds. And then we'll turn it over to you all to ask whatever questions you like of the panelists. Okay? So, Mark, why don't you get started? And then
Sure. Thanks,
everyone.
Anne. So apologies for the slightly delayed start. We had some road closure issues around the hotel. So I'm not going to spend a lot of time going through the slides I presented yesterday. I am simply going to recap the endpoints so that we all have a clear understanding of the primary and secondary endpoints, and just remind you of the headline efficacy findings. All right, are we good? Okay, thanks very much. The pointer is... do we have a laser pointer? Oh, that one. Okay, great. Okay, all right. So I thought I'd just remind folk that the primary efficacy endpoint in this trial was initial quantiferon conversion from beneath a value of 0.35 international units. And here's an example. So the dark line being the threshold. So in the blue is an individual who had a low value that exceeds the 0.35 threshold. That would have been an example of our primary endpoint. The secondary endpoint in this trial, sustained quantiferon conversion, post day 84, sustained through 3 and 6 months. In other words, 3 consecutive tests. And in blue here we have an example of sustained conversion, in fact at a high level, ending up greater than 4, and we'll show in the subsequent slides the effect of alternate threshold values that were used in the exploratory endpoints. Some examples of the alternative conversion thresholds, using here the 0.2 to 0.7 threshold, shown in gray. Here we've got an example of an individual in blue who converts from beneath 0.2 to greater than 0.7. And finally, the initial conversion value of greater than 4, which we've previously shown to be associated with very high risk of TB disease. Here we've got an individual converting from, in fact, less than 0.2 to greater than 4. So to recap very briefly the major efficacy findings. You'll remember from yesterday we had 134 initial conversions, or 14.4% of the entire cohort, giving us an incidence of 9.9 per 100 person years. The rate in the placebo group is 15.8%, in the H4 group 14.3%, and in the BCG group 13.1%. And you can see these conversions were fairly evenly spread with a 6, 12, 18 month time point. The efficacy for H4 using the initial quantiferon conversion primary endpoint was estimated at 9.4. It was not statistically significant by any threshold. Similarly for BCG, the estimate 20.1, not statistically significant at either 80 or 95% confidence intervals. So the secondary efficacy endpoint, being sustained conversion, we had 82 sustained converters, so who had 2 subsequent positive quantiferon tests after the initial conversion from negative to positive. This is 8.8% of all participants, 62.6% of initial converters. The rate of sustained conversion being 11.6% in the placebo group. And you can see here that those sustained converters in the placebo group seem to be clustered at a very high level compared to the BCG arm. In H4, 8.1%, in the BCG arm, 6.7%. Calculated, reversion being the complement of sustained conversion. The reversion rate in the placebo group being 25%, 40.5% in the H4 group, and 46.2% in the BCG group. And again, you can see here that in the BCG arm, you have the suggestion of reversion happening from a very high level. Also note that time to reversion, most of these reversions are happening by 3 months after first conversion. So calculated efficacy for the secondary efficacy endpoint, 30.5 for H4, statistically significant at the 80% confidence limit, not significant at the 95% confidence limit. For BCG, 45.4% efficacy, statistically significant at both 80 and 95%. And I think I'll stop there and we'll contribute to the discussion later.
Great. Thank you, Mark. And just a quick reminder that in terms of safety, both vaccines, both H4 IC31 and BCG, of course, had very good safety profiles. BCG having a high rate, as expected, of the local site injection reactions, and both vaccines being immunogenic as well, as you heard yesterday. So let me introduce my fellow panelists here, and then we'll start with the questions. Mark, you already know, Gavin Churchyard is founder and CEO of the Orem Institute and a very internationally well-recognized clinical investigator, both in TB and in HIV, with wide experience of both vaccine and drug trials. On my right is Carl Diefenbach, Director of the Division of AIDS at the National Institute of Allergy and Infectious Diseases of NIH. And all the way to the right is Sanjay Gurunathan, Associate Vice President for Global Clinical Development at Sanofi Pasteur. And Sanofi Pasteur is the sponsor, of course, of the vaccine. Aeras, Amanda Ginsberg, Chief Medical Officer at Aeras, and we were the trial sponsor for this trial. So why don't we get started? Sanjay, maybe I could start with you. Do you want to share and take the microphone? What you see is the key takeaways for the H4 IC31 vaccine.
So when we step back and look at the results, I think a couple of things emerge. I think we have to be aware that this was a first of its kind study. And the way we designed the study, the assumptions that we used to design the study were, the best guesses we could exercise at that point. We used those assumptions to set performance parameters around the study and design the study. It clearly emerges that there are lots of important scientific leads that we can actually pursue from this study. And there's a lot of important learnings that we need to further interrogate and explore as we move forward. Particularly for H4, I think the news is exciting in the sense that it's the first time we've seen a non-zero signal for a subunit vaccine. So that's exciting by itself, although the strength of the signal wasn't at a level which got us very excited, but nevertheless, conceptually, I think it was a huge step forward in that we could now demonstrate that using a subunit vaccine, we did see a limited biological effect. On the other hand, I'll turn it back to you, Anne, on your take on how BCG performed.
So we will ask each other questions, and then others amongst us can chime in as well, of course. So I think from our perspective, the BCG revaccination results are really exciting because they've done a few things. They've helped to confirm that the POI trial design can be a useful tool for making decisions about vaccine candidates and for understanding whether a vaccine is doing something biologically important. We think the results should be confirmed in a prevention of disease trial now, because in the end, of course, preventing disease is what we need any vaccine to do in order to impact the epidemic. But in this case, I think we could do a couple of things. We could confirm these results in additional settings. I think it's really important that in this case we were studying uninfected, high risk for infection and for disease individuals, right? Whether we'll see exactly the same findings if we move to a setting with a lower force of infection, for example. These adolescents in Worcester, as you heard, are converting to quantiferin positive, becoming infected at a rate of about 11% per year in this trial. So that's an extraordinarily high rate. And it would be important to know whether the vaccine BCG works equally well in more moderate settings as well. But importantly, we need to confirm these POI results, but we need to expand them to see if then they translate into prevention of disease as well. So I think it's really exciting for the field that there's the possibility of using one of our current tools better while we're developing even more effective vaccines. For us, that's the key takeaway, but certainly interested in others' thoughts.
So I think, I'd agree. The POI signal, I think, is a tremendous advance for the field. We've demonstrated that we can demonstrate an efficacy signal with a prevention of infection design and do so in a high transmission area, and that's very important because the question has remained ever since the MVA85A trial, whether in fact such a setting was a high bar for a vaccine. So we've demonstrated in a high transmission setting that we can demonstrate efficacy for a vaccine, and I think that's a major step forward.
Yes, I think for me what's important is that this trial has important differences from the previous BCG vaccination trials done in Brazil and in Malawi. And just to contrast them, because it will be important to understand why here we've got a positive signal and we didn't see that in the other two trials. So in Brazil, this was a large cluster randomized trial with passive follow-up involving two cities, Salvador and Manaus. And overall, they didn't see an effect of BCG in that trial. They followed up for first 5 years and then extended it for 9 years, but then did see an effect in Salvador amongst children. And so the signal may have been lost because they were just, they involved everyone, including people that were infected with TB. In Malawi, that was done in the Karonga district in northern Malawi, and they randomized about 50,000 individuals to BCG, but again vaccinated both infected and uninfected individuals, and so didn't show any effect of either primary BCG vaccination or revaccination. Even though it was efficacious in preventing leprosy, it didn't prevent TB. So I think what's unique here is we've really focused on individuals that are at high risk. We've been clearly able to demonstrate the efficacy, and that this now allows us to move into the next stage. And I agree that we need to do a confirmatory trial in other settings. And the hope is, of course, with low transmission, it may be even more efficacious, and that should be embedded in a large phase 3 trial. From the correlates of protection, I mean, this is the first time we have a trial that we can use to look at correlates of protection, but it's still a small trial. And so unless we do the confirmatory, we may not have sufficient power to look at correlates, but certainly we can start looking at correlates.
Okay. Mark, maybe you'd like to kick off some thoughts on what we've learned about POI trial designs and how what lessons you take to the next POI trial.
Yeah, so I think the most obvious lesson is about endpoints. You know, we powered this trial based on the initial conversion endpoint, and I think as Barry raised in one, you know, his early presentation, if you design a trial with only one endpoint and you guess wrong, then you're in trouble. So I think we were particularly lucky that we saw a lot more quantiferon conversion endpoints than we expected. And because we saw more endpoints, we had more sustained conversion endpoints, and that's where we saw our signal. So I think the lesson going forward is that we should be powering future trials based on a sustained conversion endpoint. Still analyze, obviously, initial conversion, and we saw a signal at those high conversion levels greater than 4. So I think that is still important, but powering future trials based on the sustained conversion endpoint.
So I think, and a couple of things to highlight. We talk about replicating this study in a different setting, but I think we must also recognize in parallel the unique features of this study. The first being that this was a high-intensity community setting. And we often talk about household contacts, we talk about other cohorts that are reasonably well characterized or well characterized. So as we think about reproducing these results, I think we need to keep the target population into perspective. The second thing is the endpoint, as Mark was alluding to, is intricately linked to the type of assay you use, right? And in this case, we use quantiferon gold. But there are other options out there also. And I think it's important as we think about replicating this study, whether in a similar POI design or a POI POD type of design, whatever the design may be, keeping the assay constant or similar is going to be really important in the way we enforce endpoints for similar studies in the future.
So I'd like to say a few things about that exact point. I come from the HIV vaccine field, and I think that understanding the fundamental properties of your biomarker are an essential component of understanding how your vaccine performs. So this idea of doing anything to change the variable going forward in future POI studies doesn't make a lot of sense unless you include this as your standard. I think some significant work needs to be done to truly understand what it means to lose your quantiferon signal. Does that truly mean that you are essentially disease-free or cured? So by analogy, I think about the biomarker we have in the HIV field, and that's viral load. You can have a zero viral load, but that doesn't mean you're cured. It just means there's no replication. So I think we have to spend a significant amount of time understanding what the biomarker is, and then try to also understand how that biomarker plays with the measurable immune response we see from either the H4 vaccine or BCG. Because you can measure immune responses, but you do not necessarily know you're measuring the protective response when you perform these kinds of measurements. Secondly, I think one of the concerns, if we think about the studies that were done in Brazil and in Malawi, was the time. And yes, it was a community randomized trial, but it took 9 years to collect all the endpoints. So a concern based on the rapidity of what we're seeing in this study, that we may not have a durable response to BCG revaccination. We need to really understand how durable this is and whether or not we see an effect a year or 2 or 3 down the road. Lastly, we've heard a lot at this meeting about other whole cell bacterial-based vaccines, and will they have a similar biological mechanism of action as BCG revaccination? So if we're going to go with the principle of changing one variable at a time, let's not change the assay. One thing you could do is add additional arms to a POI study to then generate additional data on other vaccines to see if you had the same sort of quantiferon response. So I think that this is a very exciting result, but you're as good as your endpoint, and as good as the biology behind your endpoint, and I think some significant amount of work really needs to go into truly defining what is going on. As you notice, not everybody went to zero. They sort of stayed in a low level of interferon susceptibility. That's like a low level of burden of disease potentially, but at what point does that break through and they burst up and go back over a level of 4, indicating potentially loss of immune control? So again, I think it's a great start, and I think if we take this in a stepwise fashion and move accordingly, and be confident in the answers at any given stage, good decisions can be made, and quality science will prevail.
Tom, did you want to come up and use this one? No, here we go.
So I completely agree. These are excellent points, and I mean, maybe I can ask one of you to speak to. So 2 things have already changed. The first is that SSI BCG is not available anymore, so the product that was used. And secondly, the Quantiferon Gold is no longer available either. So now we have the Quantiferon Plus, which is by and large the same, but it is a little bit different, and and perhaps we need to speak to that and just kind of get your opinion of it.
Yeah. So in terms of the BCG product, I know some folk in the field would be divided as to whether different strains of BCG truly have measurable differences in efficacy. But the reality is that the only way to be sure that we will see the same signal with a different strain is to repeat it in a trial. Otherwise we're just speculating. The same goes for Quantiferon Plus to a certain extent, but I think so with Quantiferon Plus, as Gavin was alluding to yesterday, I think we might expect greater sensitivity, which then might affect our ability to detect initial conversion. How that then affects our detection of sustained conversion and reversion, again, only time will tell.
So in terms of Quantiferon Plus, because it has the second tube with CD8 specific antigens, it's thought to be more sensitive, and in the initial studies done in Japan and Australia, it seemed to be that way. But a study done in Zambia, looking at the sensitivity, showed that it was had increased sensitivity in HIV infected individuals, but much less so in HIV uninfected individuals. And in amongst South African gold miners who are HIV uninfected, absolutely no difference between plus and gold. So again, the assays change, and we actually don't understand what it's really contributing. So we do need to understand that. But it may continue to change because Cryogen are now also developing a lateral flow assay for plus. And so if they stop commercializing doing it in the tube side, we may not be able to get a quantitative result, which would be a real problem for the trial. So we need to make sure that they continue to manufacture the tubes in the current format so that we can have a quantitative result. So this trial was done, of course, in HIV uninfected individuals, and that was an important difference that I've neglected to mention between Brazil and Malawi, where it was the early stages of the HIV epidemic, and certainly in Malawi, where there were excess cases in the BCG arm thought to be due to be HIV-associated TB. Clearly, we can't do this in HIV-infected individuals because of concerns of BCGiosis. But programmatically, that will be very challenging. So as we think of going into prevention of disease, we do need to think through how we would operationalize this.
So I think the the HIV, I mean, as we all know, the HIV epidemic is changing, and with the increasing number of individuals going on to ART, it is quite likely, in fact, that the risks of BCG vaccination of HIV-infected people will be much lower than they have been historically, and I believe that that may be reflected in the upcoming guidelines for use of BCG in HIV-infected people. So perhaps that might not be as big an implementation consideration as.
But then in the follow-on trial, we need to include such individuals.
Marcel.
Could I speak to Tom's question about changing two variables, which was raised by Carl's point about going forward? As I understand, if you were to do pragmatic studies, you get the BCG that's in your fridge and you get the quantiferon that's in your lab. But if you're doing a controlled RCT where you want to ask a question, what is our capacity to get a batch of the BCG Danish to confirm with the same thing? And what is the capacity to get QIAGEN to make the old-fashioned thing rather than leaving us what's available in the future, lest we end up 5 years from now having changed 3 variables and had a different result? And I don't know the answer to whether either of the old products can be available in a designated trial format.
So I can try to take a stab at least at the BCG question. Maybe Peter wants to address it.
So I think
Let's get you a microphone because this is being recorded.
So this is obviously completely separate from my business now, but I think it will be available. That's my understanding. I mean, they've said, in 6 months' time, they've said that a few times by now. So I'll not give any time indications, but it's my clear understanding that they've solved some of the problems and are ready to start providing BCG again. But precisely when is not clear, and don't blame me if they fail again. But that's my understanding at least. And concerning quantiferon, it's my understanding there that you still that you can get the normal, the one we have used for years ago in the US. I think they've never changed as far as I understand.
Sorry, I think Dave Lewinson back there has an answer to this question. We need a microphone for him.
It's kind of a simple answer. I think they're going to be doing the Quantiferon Gold everywhere quite shortly, including the U.S. But the TB1 tube is more or less the same as the old tube. So I think that does address that question, and Gavin, I think you had made that point. The quantitative part is much more important, and I think you probably saw that nice paper from Elisa that really worked very hard to make sure the quantitation was robust. And so I think it would be a huge loss to lose that. And I think we saw how important those quantitative values were in understanding what reversions meant. And so that would be lost if it went to a lateral flow assay.
Thanks for that. Tom.
Two questions. One is, it is going to be a great tool, even with variables, to rank order things if you can do multiple arms at once. So 2 questions. One is, do you need to look for 3 consecutives, or did almost everyone that had a second consecutive stay up, which would gain you 3 months in terms of deciding when to drop something out? Number one, that's the first question, because it looked like there were very few people that went down if they stayed there as a second. So do you need the 3rd test? Then the second question would be, it's totally correct to drop out people out of the first 90 days. On the other hand, if BCG did have effect of sort of clearing out an incipient infection, you may have seen something in those people that you dropped out over the first 90 days. Did you follow them to see if the dropout rate was different in the BCG arm versus the placebo arm or the H4 arm? And in the future, I think when those people drop out, because they went up, you need to follow them to see if they also revert from the early phase. You need to understand that early phase infection. I think that might have been something that would have been interesting to have included in the trial.
Yeah. So we didn't, those who were positive at 3 months were not followed with quantiferons after that. So, and I'm trying to remember, is Catherine here? Yeah.
Yeah, I mean, my recollection is that the split maybe wasn't exactly the same, but it was pretty similar across arms. So there were more, I think, than we had predicted for the rate, but it was fairly even. So.
And what was the second part, Tom? Oh.
Yeah, my recollection is there were no more than 1 or 2 people who reverted at 6 months. It was almost every single person who reverted at 3 months.
Can I speak to that?
Yeah, please.
Yeah. So I think this is a really important question, the timing of the repeat testing, and I think if you think of the historical studies that have been done with quantiferon and compare it with the one that we've done, serial testing at frequent intervals picks up this phenomenon of conversion and rapid reversion. If we had been testing a 2-year interval, we would have detected a small number of individuals with recent conversion, and then the cumulative sum of everyone with sustained conversion up until that point. So I think it's the question for future trials is, are we interested in detecting those rapid reverters, or are we interested in detecting the sum of the sustained converters over time?
I think one of the issues the field's going to absolutely need to grapple with is how to measure durability. I think with some vaccines it's pretty straightforward, but when you're dealing with this kind of a biomarker assay, I think it's not a straightforward measurement to get to durability.
I mean, I think this is a really important point, especially for the way we understand or we interpret what's actually going on. So if we had only measured quantiferon at the end of the study, then we wouldn't have picked up that these people got infected or converted. They would have looked like they just never actually converted, which means they wouldn't have reached an endpoint at all. But the other thing is that I think it helps us to understand what humans are capable of in terms of the immune response. So if our interpretation is that a conversion followed by reversion actually represents infection and then somehow controlling that infection or perhaps even sterilizing that infection, I think that is really quite an interesting result and something that many people in the fields will probably still not consider to be true. And I'm not saying that the data proves it, but it certainly points in that direction. And I think this is quite different to what people expected. So I think understanding the biological effect that's going on will also be very important.
I think you bring up an important point, Tom. So one of the issues that I sort of don't understand with the data is how to deal with these reverters, right? And how, in the context of really the endpoint that we're talking about. Right? So clearly, if you were to repeat such a study, I would not use the same primary endpoint. We would go for sustained conversion as your primary. Whether it requires 3 samplings or 2 samplings, I think that's an important point that Tom had raised that needs to get resolved. And I think we need to look at the data and see if we can resolve that. But then, what do we do with the reverters? Do they represent just a mirror of the sustained converters, or are they a separate and distinct population? In other words, would you consider your sustained converters and your reverters as a composite endpoint? And does that make a difference? And I don't know. I mean, these are important questions that need to be answered because you have the potential to lose a very valuable signal if you don't think about your endpoint carefully.
So if I can pick up from that. So in the adolescent study that Mark and Tom did, that in the quantiferon negative individuals, a similar proportion had the correlate positive. And one wonders whether they were individuals that may have been converted and then reverted. And it's the point that Carl was making. What does it mean? Is that the group that have a they are at lower risk, but are still nevertheless at a higher risk than those who never converted and reverted. And so in the phase 3 trial, I would agree that we need to follow them out as a separate group. I wouldn't just say, well, that, you know, don't count them. We need to watch what they do, and if they are at increased risk, but even though it's lower, we need to document that and understand the biology, because they themselves may need additional interventions. You know, they may not be protected. That may actually be increased risk as well.
The notion of understanding the biology of these individuals is the essential basic science component that's going to really make or break our future here. The idea, I think we have to go at this without prejudice of what the fundamental underlying immune mechanism is. It could be innate, it could be CD8, it could be CD4, it could be NK. But I think that we need to make sure that the team has the opportunity to build a set of assays, validate them against placebo samples or other, so that then these kinds of correlates of analysis can be done in a way that further refines the question to where you define a correlative risk that could then, in addition to using the quantiferon, provide another marker that you would look for in an expanded study. I think this can all go on in parallel with planning additional studies and in using the other ongoing studies as feeders for samples. I think in the HIV field, we had a study called RV144 that had 31% efficacy. It was pretty terrible level of efficacy, not enough to license, but through this kind of analysis, we were able to identify 3 correlates of protection and 1 correlate of risk that promoted the degradation of any signal. And so with that kind of effort, again, in parallel with other forms of vaccine development, it really put the HIV vaccine field on much firmer footing, so it's not sort of a wing and a prayer. So we really have this opportunity here to build on this while we do some additional studies.
Put your hand up high so we can see.
Thank you. I think it's fascinating. But I'm interested to know what samples you already have banked that you might do some correlates or biomarker analysis on. Because I think, well, I don't know if Stefan is here, but I mean, I think he may tell us later about how there are signals coming out not only that identify potential progressors towards disease, but also there are some metabolomic and gene transcription signatures that are beginning to look like they are an indication of sort of challenge or initial infection. And so I think it'd be very interesting to look at those in the converters and reverters.
So briefly, we have collected specimens throughout the trial from all participants, which are banked. And I don't remember exactly what time points, if anybody else does chime in, but okay, come. Great.
So we collected PBMCs from all the participants, but we only collect them up to 12 months. And we did not collect them when we repeated the quantiferon test. Mm-hmm. So we will not have samples to look at what happens in those who revert. Although at those time points we did collect RNA, so we can look at the correlate of risk signature to answer your question about whether that signature is still higher in the reverters compared to perhaps those who never convert. And then we have plasma and serum at all these time points, and also RNA. But I think the focus was to measure vaccine-induced responses before the endpoint occurred. And I believe due to budget limitations, we were not allowed to collect more samples after the endpoint was reached, so that's a pity.
Thank you. Peter.
So I just want to make
Peter, I just want to add one thing before, just to finish the thought a little bit on RV144 in terms of the correlate analysis. What was so fantastic and peculiar about the way we did the correlate analysis was the way the model was set up, actually asking for help from premier labs around the world, right? And it was not restricted to one lab or the other. Proposals were sent out, and the best assays and the best investigators were chosen. And we recognized that these samples were worth their weight in gold. And we had a setup and a process on how to govern testing. And I think it's important as we think about these samples that are banked, how we actually go about testing them. So the process is equally important.
So just to let you all know, there is actually a process being put in place modeled on the RB144 example. The Gates Foundation, and Karen may want to talk to this, has provided a planning grant to Georgia Tamaris, who helped lead that particular effort. And she has gathered a working group that is will try to make recommendations about which assays, et cetera, and set up this kind of RFP process so that labs that have assays that they think would be appropriate can make themselves known and sort of apply for samples. There was something else I was going to say about this. I've lost my train of thought. Does anybody else want to add in on that process? Well,
No.
Okay. Go ahead.
So I just want to make the point, and it's already been touched upon, that I mean, this is a unique part of the overall population being the quantiferon negative, and in the quantiferon negative, we would expect BCG to have a relatively high chance of replicating and not being blocked because the immune responses are relatively low. I mean, the chances is that, or the risk is that we would end up in a different situation and with a different ratio of efficacy between a subunit strategy and a BCG strategy in the quantiferon positive, where BCG would be blocked by the strong immune response by NCBI, and where all the animal data would suggest that the subunit, which are not subject to blocking, would have a relatively larger role to play and a larger chance of working. I mean, and I mean, it's just, I mean, the way we put this trial concept together, I mean, it's difficult to see what we could do to go around that. But if we somehow, when we move on, could convert this plus minus to really a quantitative readout, that would also allow us to measure the relative impact of already positive quantiferon levels in quantiferon positive, so that we may be able to, modulate this overall response and really look at that as a potential readout for the future, because I think we need to focus on the quantiferon positive part of the population also.
I really want to stress the need to be agnostic about potential mechanism, because while we believe that gamma interferon is actually measuring something essential, it may be part of the signal or irrelevant, and it may be a secondary byproduct of just inflammation and response. I really think that's why I think it's critical that we take this approach, break down the assays, look at dynamic range, and pick some up. At the same time, as you design a study, the next study, one possibility is take quantiferon positive individuals, enroll them in the same sort of revaccination, stratified by what their level was, and then do these kinds of analyses. I think there's some very good tools at our disposal as a field that we can use if we think through all of these necessary steps. But let's try to keep in our mind that we all have prejudice. I think that's a good thing. We have ideas about what the mechanism will be, but we really need it to be a true discovery effort that is statistically powered.
And just a reminder to your point, Peter, that there will be data coming out this summer on a quantiferon positive trial of a subunit vaccine, different vaccine, of course, but I think, again, we need to keep an open mind, and that'll be very interesting to see the results of the M72 trial. So, Tom.
So, logistic questions. In order to do exactly what you're saying and have sort of lack of diversity among studies, it would mean that a central funder with lots of funds would be able to set up sort of a rolling study, which was one of the original thoughts we had of just continually enrolling people and putting arms in and then doing overlapping placebos. This is part of the original design. So 2 questions. One, I'm a new person. I come in, I have my vaccine. Is either the Gates Foundation or the NIH going to commit to doing a high quality system so that we don't get these one-offs all over the place that have variable conduct and variable kind of settings? You need sort of a something that's more steady, that's constant, so that you get the same endpoint. And the second thing I think probably the audience would be interested in hearing is, what did this cost, and what would it cost for me to put 350 people into an arm? Because I think we should be transparent on what these things cost.
I'll be completely blunt with you. The issue is, what is the question you're trying to answer with your vaccine? And I think it has to remain a hypothesis-driven activity. It is up to the field to coalesce around a structure that's going to make this work. RV144 analysis came about because there was a willingness by a group at Duke to take this on. And yes, they were financed, but ultimately a lot of people put a lot of free time and money into this. That said, we will always fund meritorious ideas, but what is your preliminary data? What is your hypothesis? This is not going to lead to a VTN-like structure for TB vaccines. Let's be honest about that. But there are opportunities here to grow this field on the basis of very strong science, based on some of this data. And I'm actually curious about what the 350-person arm would cost too. I'll turn that one to Ann.
So this was a 3-arm study, and I'm not going to give exact cost for this trial, but I can say in general, a POI trial conducted this way with the biobanking involved, not the correlates analyses afterwards, but just the banking, is on the order of $10 to $15 million. So that was for 990 subjects.
Can I add to that? So we should also remember that this trial was powered on initial conversion, and if we're going to do future trials powered on sustained conversion, that's going to up the ante significantly.
So one comment I have back here. I mean, this was called a prevention of infection study, and yet it turned out to be more about control, or maybe prevention of progression, at least with regard to what we've measured so far, which I find remarkably concordant with what we've learned from our animal models. That is, we really have any fantastic examples of an in vivo vaccine model that actually prevents infection. What we do is we modulate infection and we prevent its progression. So in many ways, this is an example of a human study that's actually quite a ways ahead of our animal modeling. And so it's a really interesting and fantastic design, but it sort of highlights the need to begin to develop models of non-infection or the non-take that we might actually come back to a true prevention of infection study.
Can we get a microphone?
Thank you. The fact that people remain at risk of infection over time brings a layer of complexity. I come from the malaria world, where people are under constant exposure, and children in studies or adults often have repeated infections. And vaccine efficacy measured as vaccine efficacy against first events turns out not to be a bad proxy of vaccine efficacy against all events, because there is taking into account repeated events over a longer period of time. And that's interesting for malaria, because reducing the number of episodes is something important. Nevertheless, it is not a good proxy or marker of measuring vaccine efficacy, considering the number of people who remain free of any episode, which is what matters for a disease like TB, which is more, where what we want is to prevent having any TB. So what happens during subsequent infections is really important. My question, I was interested to know whether you had second episodes of conversions in the study. I would imagine we're entering very, very limited numbers here, and it's going to be difficult to interpret. But approaching that and the question of durability of protection is going to be very important.
So I think there were maybe 11 people or so who had gone from positive to negative and then back to positive. But I mean, to me, it seems different from malaria in that you're dealing with disease episodes, not necessarily people suddenly becoming parasitemic again. You know, it might be they've hit a certain threshold where they are showing clinical disease with malaria. So to me, there's a bit of a difference there personally. But I would say too, as an aside, I think that was one of the reasons that we were interested in looking at some of the other quantifier thresholds for our analyses, particularly from some of the work that Satvi and others were doing that showed perhaps a greater specificity or larger association with a higher risk of disease if someone were experiencing a higher quantiferin level. And so I think that whereas the going from positive to negative and back to positive may be important, I think it's also useful to see is the manufacturer's threshold actually the most appropriate cutoff to be using, and you know, if not, maybe there are others that would be more meaningful for assessing potential disease.
So, question to Mark. We've used just quantiferon. Clearly, using a tuberculin skin test is an immunological intervention in itself. But we know from other data, particularly amongst household contacts, that there's discordance, and you know, someone may be quantiferin positive but TST negative. So, what is the phenotype of being infected? Is it being quantiferin and TST positive? And does that have to be sustained? Because again, in household contacts, of those who have initial conversion, about a third occur quite distal from that first exposure. You know, they occur over a period of a year. And so I think we still haven't, you know, we've used quantiferon just because it's easy and we're not interfering with the intervention. But, you know, clearly there are others that are quantiferon negative but TST positive, and would we really say that they're not infected?
So this came up in one of the poster discussions earlier this week, and I think what it comes down to is, you know, if you're using more than one test for the same thing, then you start to muddy the waters considerably if you're using TST and quantiferon. But you need a gold standard, and the issue is in, you know, TB endemic populations that are BCG vaccinated, and particularly where the intervention is BCG, you know, it makes it very difficult to use TST as the outcome. So in terms of our study, quantiferon is the endpoint, lacking a better one. Yeah. So one thing we haven't talked about in planning for the future and next steps is in planning towards what we think is the next step, which is a large prevention of disease trial, how we get there and lay the groundwork. And I think, you know, Richard's sitting in the front here, modeling studies of understanding the impact of prevention of disease by BCG are going to be key to that effort.
Yeah, I as a modeler, I totally agree with that, Mark.
I thought you meant. Yeah, sorry.
We discussed it last night, and there's work, I think, which doesn't involve transmission models, just thinking about the trial design, which are much more simple and just think about the kind of power and duration and that sort of thing. But then there's a second step where you start to think about transmission, about what the impact might be on the epidemic. My first question I asked you was not about that, though, wasn't it? It was about setting expectations for the POD. And I'm going to say the question I asked you yesterday. If you were funded tomorrow for your dream POD, when would it be? How long would it be before we were sitting here again listening to your pop star talk yesterday with a POD result?
Yeah. I mean, I think that's a really difficult question to answer because we don't know what what limits we need to set around durability or our ability to assess durability, particularly for prevention of disease following infection. I'm thinking it would be at least 5 years study, like if not longer.
So when I was training, I was taught that as soon as you study a problem, it goes away. And it seems you had 0 cases of active TB. Could you remind us how many people years of QFT conversion is the denominator for which you had 0 events? Because that might help us to also calibrate our expectations.
Yeah. So I'm sure Catherine can give us the exact person years, but I can give you the estimation. So from our projected number of cases, the 95% confidence interval around the projected number of cases was 1 to 13. We expected maybe 5 or 6. We didn't see any. Remember that this is a slightly de-risked population because we excluded individuals who had a household contact from entering. They clearly were not entirely de-risked because there's a lot of transmission happening in this population. But that might account for the fact that we didn't see any cases. And there are no HIV-infected individuals here. Catherine, do you
did some rough sample size calculations for what a POD study might look like. Rough sample size calculations on what a POD study might look like. And we changed some assumptions. We included in that calculation a proportion of quantiferon positive people. And, of course, a large proportion of quantiferon negative people. And roughly, it comes to about 30,000 subjects, give or take some. And so you can already see that if you now are going to progress to a POD study, you're going from roughly about 1,000 subjects in this study, which was essentially designed to be a triage study, to, almost, 30 times more.
Is that 30,000 for 3 arms?
Two arms. 2 arms.
15,000 per arm. Thank you.
Roughly. I mean.
Other questions from y'all? All right, well, I think we've talked a lot actually about potential next steps, but if anyone has additional comments they want to make on either types of trials or scientific questions that are important to focus on.
One thing that might be important to me to talk a little bit about is follow-up of the individuals who were in the trial and what's going to happen to them and what can still be learned from them, etc.
So I think we have a licensed vaccine and we need to take it forward in further trials. But as I mentioned yesterday, we haven't really done what is the optimum dose and route of delivery of BCG. So in parallel with doing all of these efforts, we need to see if we can actually optimize that as well.
Mark, do you want to address Tom's question, or you want me to?
In terms of?
Follow-up.
Oh yeah. So I think in terms of long-term follow-up of this particular cohort, I think this cohort is relatively small in terms of getting an indication of disease efficacy, but there's obviously understanding in terms of the immunological changes in this cohort more distant from injection. So those studies are certainly worth doing, but if we're going to do them, we must decide to do them fairly quickly.
Are you studying the correlates in this group?
That would be a good idea.
I think using this cohort as at least to try to get a read on durability. You're a year or two in. I think that would be very useful.
So one of the things I wonder, we talk about having some sort of confirmatory trial where we minimize the number of variables that we change. And I know the strain of BCG and the type of quantiferon test have been raised. I think one of the other things that has really stood out to me in going through this trial and going through the results is people being very, people being very hung up on significance level. And, obviously the trial was designed with a one-sided alpha of 0.1, which I know isn't standard. However, there were very reasonable justifications for why it was done that way. And I think if we were to potentially, redo this trial and go with a more traditional alpha level, it's going to have a huge impact on the cost because you're going to have to increase your sample size so much. And I think coupled with that, I think we'd be unable to really reduce the number of quantifier tests because I think Tom mentioned earlier you would lose some endpoints because people may be reverting and you never have the opportunity to catch those endpoints. So I guess I'm just trying to think of other things that may come into other considerations that may come into redoing a trial. Excuse me, especially knowing that we're seeing modest vaccine efficacy with H4, for example, and have seen other types of diseases where prevention of infection perhaps is more modest than prevention of disease, and perhaps would be seeing an even more meaningful result if it were some sort of bridged trial of prevention of disease.
Is there a question buried in there?
I'm just wondering, I guess I'm just wondering what other types of considerations would be, types of considerations would be made for a next study if it were confirmatory, given some of the issues, I think, in terms of how people perceived maybe an atypical confidence level, for example, and some of these other things that may contribute toward vaccine efficacy.
So I just wanted to ask sort of the higher level strategic question, and this is really to all of the panel. How do the results of this trial change the way that you think about the TB vaccine field, if at all?
Very exciting, and I think these results should be followed up. It's not often you get a positive signal, and it's a travesty if you don't follow up the positive signal. And the two positive signals we got in this was that the subunit vaccine showed a non-zero result, albeit modest, and the BCG showed reasonable results with statistical significance that warrants further pursuit. Now, exactly how that's going to happen, there's a variety of options. We heard today that let's not get carried away, let's repeat the experiment in a different setting to make sure that the results are holding up. That's one pathway. And the other pathway is to say, okay, the prize at the end is the prevention of disease study. And that's what's going to give you the greatest public health impact. So we should move forward, at least with BCG, in that model and see how it plays out. So I think the field needs to actually debate which direction we want to take.
I would agree with that, and I would add that we really have an opportunity to clarify what we actually just saw. And I think as we go forward, then depending on what the answer to that is in terms of durability, what the nature of the response is, the ability to trigger said response by either a subunit vaccine with additional antigens in it or some other vaccine modality will really help define the future. But you don't have a positive signal very often, as Sanjay said, and let's take full advantage of it.
So I just want to mention that that we've developed this ASAT-free ICRA together with SAT. We have validated this, so it's actually available for vaccines based on ESAT. But interestingly, in addition to that quality, and that's the reason why we developed this, I mean, it's adding new antigens that can be used to increase the sensitivity of the existing ICRA. meaning that we could use it as an exploratory assay when we continue these efforts to increase sensitivity of the overall readout. So that's one point. And then the other point is to Gavin's point about the TST. I mean, in addition, we have developed this skin test based on the IGRA antigens. And this skin test is far progressed. It's been through all the clinical phases. It's ready for manufacturing, basically, and we're looking for a partner for this, which we will find. It's a niche product, unfortunately, so it's not been that easy, but it's we will find a good partner for that. And that would provide a skin test, specific skin test. I mean, and given that you don't want to intervene, as you say, it would still be interesting in some kind of bridging studies to see how it would work, because it's not identical. I mean, you know, it's very, very similar, and it has the same sensitivity, but you still find discordant individuals that would respond either to one or the other.
All right, I think we're almost at the end of our time. Any burning questions left before we thank you all?
Yeah. So Anne, maybe we could give Gavin and Mark a chance to answer my question as well.
Yes, I agree. I think this is very, very exciting, and having been in the HIV vaccine field for many years and sort of been through these valleys and highs, having a positive signal is really, really what's going to galvanize the field. And so we've kind of followed behind what's happened in the HIV vaccine field. We need to grab this opportunity with the same enthusiasm and determination as we did in the HIV vaccine field and move forward. So.
I agree. I think this is a fabulous result. And I think we now have a tool. The POI design it needs to be validated, but we have a tool with which to evaluate new candidate vaccines. And that tool gives us some indication of biological effect.
Okay. Then I think it just remains for me to thank the panelists very much and to thank all of you for your interest and questions and comments. Thank you.